We start this part just after Dr. Sparrow finished with her comments, and as Dr. Strano-Amore is coming up to the front.
Thank you both for covering those important and very interesting topics. With genetics in these past few hundred years, we as a species have made great progress, from the days where the genome was first discovered. First step was trying to grasp how DNA carried the information, that is the blueprints of who we are, and how it changed from one generation to another. We looked with wonder, the processes that made new life in each generation, from different parts of the old. Traveling through enough generations, we could already see how different combinations of old also somehow incorporated new elements, and evolved into new life forms, and eventually to who we are today. The first attempts we had at trying to control this process met with disaster. Cloning led to premature death, massive health problems, infertility, and in short, a big mess. Knowing that nature seems to cut and paste parts of the genome from generations past with ease, led more than one scientist to wonder with admiration and envy at how much better nature is with genetics, and how sorely lacking we were. Over the years, by studying both the processes that nature was using, and the many abysmally bad, and some unconscionable, tests that had been run over the years, patterns began to emerge. To condense many hundreds of years of research, and a vast amount of technical data, we began to see just how unstable and fickle the genome was. We also began to see a depth of beauty, elegance, and fragility in the genome, past anything that could have been imagined to that point. The genome was found to be unstable when edited or copied. It also was found to contain a lot less useless information than was initially thought. Many parts that seemed unneeded turned out to be natural built-in redundancies for very important biological processes. Getting the copies and edits right, without knowing fully the many ways in which the small parts of the genome really do function, proved to be an exercise in both futility and in creating abominations with short, cruel lives. But at least that did add to the data that ended up helping us understand the genome through these many years, the steadily increasing knowledge of how DNA works, in each of its many detailed parts, and in how Mother Nature naturally edits the genome from one generation to the next. We began to be more successful in harnessing the genome. So in short, it is both the knowledge of the data and function of the parts of DNA, as well as the knowledge of how nature naturally edits the genome, that gave us the ability that we as a society have now. You can see that clones our competitors make lead almost as long of a life as natural-born humans, and our genetically grown organs can last the remainder of our clients' lives. But everything now is, and has always been, measured in relation to a natural-born genome. Even with as much as we have learned, we still have not completely caught up with Mother Nature. With this new project, this is simply not good enough. Our new genetics need to solve the problems of editing and copying the genome, and why it leads to instability, uncertain effects, and shorter life. It needs to be sturdier than nature, and far more robust to have any chance of getting DNA that can withstand the dramatic effects of extreme long life, and a lifespan without a technical limit. While all of the other discoveries that have been made up to this point will unquestionably be an essential part of this process, the new step forward that was not possible before is a more complete use of triple helix, 3-strand DNA than has ever been used before. Since we are already born with a double helix, 2-strand DNA, incorporating newer 3-strand technology with an already 2-strand host body would prove to not be easy. Since we are moving from an old body to a new one, for the first time, this becomes a very realistic possibility. The whole body can be designed from the ground up, with more robust 3-strand DNA, and since it will always have been from its creation, a triple helix DNA, there will be no problems of compatibility with the old. Now we have covered somewhat the how it is possible, I dare not forget to cover the why. Getting back to the core problem of DNA and genetics modification, we see the instability and longevity of edited and artificially copied DNA. The third strand acts not so much as a way of incorporating more information, but rather a way of stabilizing the other two strands. In our uses of it, it makes the new DNA less likely to have genetic drift over time, and more stable and resilient to edits of DNA where we cut and splice new DNA into the old. While we like who we are, the DNA going forward does need to be modified somewhat to withstand the effects of extreme long life. With the telomere example, we can now, with the stabilizing effect of the triple helix system we have designed, along with the many other techniques perfected over the years, cut the key parts of the DNA from life forms that do have natural telomere regeneration, and paste them into the new DNA that we are designing. As much as we are emotionally tied to the DNA that we have, it is a somewhat random mess of DNA that has come down over the years, from this and that animal, and human ancestors. In order to move forward, we need guided evolution, and a new direction, based on science and decisions, not just based on what had made us more likely to either kill better, or procreate better, than anyone else around us. We can already kill and procreate very well. We need wise genetic choices, not just ones based on classical evolution. Going back to the triple helix model we are using, I would compare it with kids building blocks, or even with more advanced construction techniques. Without a support structure, bricks and components of structures can be far too wobbly to stay put. They fall apart easily, and large or intricate structures are either very difficult or impossible to build. With support structures, we can build marvelous creations that often need little or no extra support after they are completed. The classic arch is just one of a great many examples. The three-strand process, when added to the wealth of techniques we have already perfected, is just enough to push us over the edge in our ability to create DNA that is more robust than natural, and also more able to incorporate genetic repair and modifications that allow for a longer life. In short, we now have enough tools to create and modify three-strand DNA that will allow us to design our new bodies to weather the long harsh effects of time. We will design the new bodies based on the DNA of each one of us individually, so as not to change who we are as individuals. When trying to add a more robust DNA, what you will notice is that in the new body, you will need to exercise less, you will be less sick, and you will not age as quickly. Yes, we will still probably have to repeat the process of changing bodies again at some point in the future, but it will not be for a while, if we get this right. I have been advised that next on the agenda is our newest member here of this group, Dr. Abscondam. The question of our security has been, and will continue to be, very closely guarded, but here to explain it more is our new resident physicist.
To be continued.